Hi everyone, today we're going to look at different types of biological molecules and more specifically the types of bonds that they form during the process of polymerization wherein monomers turn into polymers. So let's get right into it. Right, some key sorts of information that we need to know prior to discussing which bonds are involved for which types of biological molecules is covering the meaning of monomers and polymers essentially and what happens in polymerization. So monomers essentially are extremely small units, and these units are repeated in order to form what we call a polymer. And a polymer is essentially a chain of monomers. And one thing that we really need to highlight here is that a polymer is a macromolecule, and a macromolecule is what we essentially define as a really big molecule. So this generally consists of over 1,000 atoms, and therefore it has really high molecular mass. And one thing we need to know is that all polymers are macromolecules mostly, but not all macromolecules are polymers. And the reason for that is because macromolecules may not necessarily be composed of repeating subunits. For instance, if we look at lipid, it's not composed of repeating subunits necessarily. But so therefore, macromolecules are not always all polymers. But for polymers specifically, we can have two types of polymers depending on the sorts of monomers that they're composed of. If they're composed of only one type of monomer, we're going to call these polymers homopolymers. And these essentially are composed of only one type of monomers. However, we can have something else called copolymers. And copolymers essentially have two or more different types of monomers. So if I had to give an example for both of them, for homopolymers, I would probably say that starch is an example of a homopolymer because it's only composed of glucose molecules. An example of a copolymer would be nylon, because nylon has diamines and dicarboxylic acid molecules. So we would say that nylon is an example of copolymers because it has 2+ types of monomers. But for homopolymers, we would say that starch is an example of a homopolymer because it's only composed of one type of monomer, which is, in this case, glucose. Now, the way that a monomer turns into a polymer is through a process called polymerization. And polymerization occurs because of a condensation reaction for these biological molecules. And a condensation reaction is wherein water molecules are removed. One single water molecule is removed as bonds form between two different molecules. And the way that this goes is water is removed and a covalent bond newly forms. And I'll show you what this means in the context of different biological molecules. In this lesson today, we're going to look into 3 different types of biological molecules, and that is going to be carbohydrates, and proteins, and nucleic acids. Okay, let's first start with carbohydrates. For carbohydrates, we will be looking at the example of maltose being formed from alpha glucose molecules. Now, when we're trying to turn 2 alpha glucose molecules into maltose, which is essentially a disaccharide, alpha glucose are essentially monosaccharide molecules. What happens is a condensation reaction wherein water is removed, and in the place of where water used to be, a hydroxyl group and an H, we essentially get a covalent bond. And this covalent bond specifically is called a glycosidic bond. And I'll draw this in order to kind of show you better how this actually looks like. Right, so we're going to have the oxygen group right there. right? So we have an alpha glucose right here. And the way that we know that this is an alpha glucose is because if we look at the sort of orientation or like the sort of location of the hydrogen group relative to the general glucose molecule, we know that they're on the same side. If the hydrogen groups are placed diagonally, we're going to call that a beta glucose, but that's something that we're going to cover in one of our later lectures. So one thing that we really have to know for this lecture is that this is an alpha glucose for the reason that the hydrogen groups are both in kind of like similar placement relative to the glucose molecule. And let's draw another one. Right, so we have two alpha glucose molecules. And what's going to happen is we are going to remove one molecule of water. This is going to go out. And in the place of where we had the hydroxyl group, the OH group, and the hydrogen, we're now instead going to have a covalent bond linked with an oxygen molecule, right? And we're going to call this sort of bond a glycosidic bond. But more specifically, we're going to call this an alpha 1,4 glycosidic bond. And the reason for that is because carbon molecules are essentially kind of labeled in this sort of clockwise direction for each glucose molecule. And therefore, this carbon right here is going to be 1, 2, 3, 4, 5, and 6 because it's a hexose. And so what happens is this is essentially carbon 4, right? And obviously this is going to be carbon 1. Because carbon 1 and carbon 4 are linked by this glycosidic bond, we're going to call this an alpha 1,4 glycosidic bond. And because it is two alpha glucose molecules, this is obviously going to be alpha. So this comes from this section right here. 1,4 comes from the fact that we're linking carbon 1 and carbon 4 together. So, right, that is the glycosidic bond, which is for carbohydrates. And in this process, we get H2O out, and we're essentially going to have one maltose, which is a disaccharide molecule. Right, so that's it for carbohydrates.